Hello, my name is Troy McCormack, and today my presentation is on the structure and function of a single synapse and how it relates to Alzheimer's disease in mice. So first off, what is a synapse? A synapse is a nervous system structure that allows for neurons to communicate with electrical or chemical signals. So here is a nice little GIF that shows a synapse taking place. Right here is the gap between the neurons, and you can see all this happening here. This is where the synapse is taking place. You have the chemicals going from this one to this one. And this is happening all over our body, all at all times. So if you can remember that it goes from 1 to 2 or from one part to another, it's just like wires in your house. You have to have it connected in order for the signal to be transmitted. So in the previous slide, we saw an example of a chemical type of synapse. But there is also another type, which is the electrical. So the difference between the two is the gap. So in the chemical synapse, there's a nice little gap there. But in the electrical one, you can see that they would be physically touching. So the electrical synapse is going to be a lot faster and much quicker than the chemical one because of that small distance it has to cover. With the chemical synapse, oftentimes this is more of a system where it creates things like a lack of certain chemicals can cause depression or can lead to behavioral changes. Whereas the electrical ones, that's more of a your quick responses to things. When you have your reflex that immediately draws back your hand from a hot object. So these synapses are taking place at all times, even when we're asleep. Um, and it's a very important aspect of our physiological makeup. So what does the structure of a synapse look like? Well, you have an axon here, and then you have a dendrite. So you have the axon, say this is the axon, and we'll say this is the dendrite. And so the synapse happens between these two. And the synapse is occurring in the synaptic cleft. You can see here there are neurotransmitters. So these neurotransmitters are these little balls here. And you can see here they're vesicles. So they would go down and then they would eventually be released, where they will go through the synaptic cleft and go in here to the receptors. These little orange guys here are the neurotransmitter transporters. So they're transporting the neurotransmitters into and out of either the axon or the dendrite. Lastly, we have these channels here. So these are voltage gated calcium channels, but they also allow for the ionic exchange of calcium and also sodium. So these allow for a change in the potential from the axon to the dendrite and allow for the synapse to take place.